Thanks, David. Hi. Well, thanks for coming to hear about some epiphyte ecology. My name is Lucy Kerhoulas, and I'm going to be talking to you today about a study that I started last winter investigating kind of some canopy ecology stuff in tall Sitka spruce trees. Let's see here. All right. So, if you live around here or you drove up for this conference, I'm sure it's no surprise to any of us in the room that, trees can get really tall. So we're kind of here in conifer country. Trees do get tall, and that's something that we're all aware of. But one thing that you might not be as aware of is that in these tall trees there's a really pretty fantastic gradient in leaf morphology. So shown here on the left are leaves from low in the crown of a redwood tree compared to on the right leaves from high in the crown of a redwood tree. And so if you're into plants and you're into thinking about plants, when you look at something like this, one of the first questions is, well, why? Like, what's driving this gross difference in morphology between, low crown foliage and treetop foliage? And a second question that might come to mind is, well, do these leaves function differently? Yeah, they look really different, but at the end of the day, is a leaf just a leaf just a leaf, or do these have different physiologies? So with what's driving this, clearly, excuse me, clearly there's a difference in light. Treetop foliage is in a high light environment. Low crown foliage is in a low light environment. So you're going to have expanded leaves deep in the crown to capture light, and you'll have more reduced leaves with reduced surface area high in the crown. But there's something else going on here. It's not just a difference in light. There's also a hydraulic component here at play. So if you imagine carrying water up 300 plus feet, a 5-gallon bucket of water or something, it's quite heavy. So same thing for really tall trees. It's quite a physiological accomplishment to slurp water up from the ground all the way up into the crown of the tree. And there are some physiological kind of consequences that go along with this. And so if we look at kind of water stress in these leaves. This is on the vertical axis, xylem pressure or water potential. So the more negative the number, the more stressed you can think of that water environment. So here on the x-axis, you've got height above ground, and you can see that at 110 meters or so, the xylem pressure is more negative than at the base of the live crown. So we call this the gravitational pressure gradient. And so for every meter of height that water has to be slurped up in a tree, the water potential decreases by about 0.01 megapascals. So what that means is at 100 meters in a tree, the most relaxed that foliage could possibly be would be -1 megapascals. And this is going to influence physiology. This is going to influence photosynthetic capacity in the treetop. So if we look at photosynthesis, sure enough, photosynthetic capacity decreases with height in tall trees. So this was a Nature paper that came out in 2004 and focused on tall redwood trees. And bear in mind, in theory, those treetop leaves, those are sun leaves. They've developed under a high light environment. They should be ready to roll as far as photosynthetic capacity goes. They should have a higher photosynthetic capacity than shade leaves in theory. But because they developed under this kind of hydraulically stressed environment high up above the forest floor, they're inherently physiologically less capable than the leaves from the lower portion of the tree. So the way that this is measured is on these cut hydros. So there's the Lycor 6400. Sorry about the blurry photo here. And here's a cut hydro. So you climb the tree, you cut branches from various heights in the tree, and then you take them back to the lab, recut them underwater. So you've essentially relaxed any in situ sort of water stress associated with their position in the tree. And then you put them under this Lycor 6400, and you can kind of see what the leaf is made of. What do they got in them? So you give them lots of CO2, lots of light, and you see what their photosynthetic capacity is. And we see that those treetop leaves, they have a lower photosynthetic capacity than the leaves from lower in the crown. Okay, so this is using redwood. Now, a tall conifer that you might not be quite as familiar with is Sitka spruce. So it's around here. We're sort of here on the north coast in the tail end of its southern distribution. It hugs the coast all the way up to Alaska. But here at these lower latitudes, they can get really tall. So it's the 4th tallest conifer in the world. Redwood, Douglas fir, and giant sequoia being tallest conifer in the world, 4th tallest. So it can get quite tall here on the north coast, over 300 feet. And there's a couple interesting things about Sitka spruce. So the first one is, yes, it's a really tall conifer, so you would think that you might witness this same kind of gradient in morphology throughout its crown. But when we look at the leaf morphology of Sitka spruce, say, compared to redwood, there's really not that much of a gradient in leaf morphology like we see in redwood and the other tall conifers. So here on the left, for comparison, is redwood. And on the vertical axis of these figures is a mass to area ratio for the foliage. So on the redwood, you can see that on treetops, the black circles, the SMA, the shoot mass to area ratio, ranges from about 200 to maybe 700 or so, a 500 spread of grams per meter squared. And then for Sitka spruce, on comparison, along its height gradient within its crown, it only ranges from maybe 1 to 200 or so. So a really not very spectacular difference in morphology in this tall crown. And so this is a little bit head-scratching, like why? How is Sitka spruce avoiding this? How are they not falling prey to this hydraulic kind of limitation that we see in the other tall conifers? So a few other pieces of the puzzle. Sitka spruce is just dripping with epiphytes. I'm not sure if it has to do with the potato chippy flaky bark is really good at capturing, spores or what, but for whatever reason, every square inch of these trees in these old trees are just covered, loaded with epiphytes. A study in Prairie Creek National Park, or Prairie Creek State Park, found over 90 epiphyte species on Sitka spruce trees. So macrolichens, microlichens, leafy liverworts, mosses, you name it, ferns, they're just all over these. So maybe somehow could the epiphytes be contributing to this lack of a morphology gradient in foliage? Maybe. The other thing, another piece of the puzzle to kind of bring you into the fold, is that Sitka spruce also produce, and this isn't unique to Sitka spruce, a number of trees can do it, but they produce adventitious roots on their branches. So these are roots aerially in the crown of the tree, high above the forest floor. And roots, oftentimes their main function is to absorb water and minerals and nutrients, that type of thing. So we have this relaxed or reduced morphology gradient in the Sitka spruce leaves. Maybe these adventitious roots are somehow uptaking a local water source from these epiphyte mats, thereby kind of bypassing the long hydraulic path that soil water would have to take. And so this kind of leads up to my study questions. Are these Sitka spruce, are they uptaking some sort of a local water source via these adventitious roots? The two main questions that I asked more specifically are if they are uptaking a local water source, we should see more relaxed water potential values in the foliage than gravity would predict at different heights in the crown. And then secondly, okay, there's not a big morphology gradient in these leaves. Is there a physiological gradient like we saw in redwood? And so the study site that I started last winter was at the Redwood Experimental Forest up by Klamath, so about 65 miles north of Eureka here. And we did some pre-dawn measurements, so this is loads of fun. One person, usually my husband, climbs the tree with a headlamp in the dark and then cuts samples at different heights, and he drops them down to me. I have my little nest of the pressure chamber at the ground and bomb away on the pressure, getting the xylem pressure measurement. So that was kind of one thing that we did and to try to evaluate this. And what we found was that sure enough, the red line there is the gravitational pressure gradient, so that would be the water potential that you you would expect just based on gravity. So if you're getting values that are more relaxed than that, so anything above that red line, that sort of is a strong indication that there is some sort of a local water absorption happening. And so for most of our data points there, you can see that they did in fact fall above that gravitational pressure gradient. So strongly implying that yes, indeed, there is some sort of local uptake of water, which would be pretty cool. The next question is, well, yeah, big deal. Like, does this affect physiology? Are these leaves, is there a physiology component here? And sure enough, when I looked at using cut hydras, the photosynthetic capacity of foliage from different parts of Sitka spruce crowns, there wasn't a nice tight kind of trend that like we see in redwood and other tall conifers. So implying that, yeah, somehow due to this relaxed water status in treetop foliage, we're not seeing that reduction in photosynthetic capacity that we've seen in other tall species. So for comparison, to remind you, there's kind of what we see in redwood, this nice decrease in photosynthetic capacity with height, and in the Sitka spruce, we're not really seeing that so far in the data, which is kind of interesting. So continuing research kind of on this topic, what I'm hoping to finish up for this winter, starting I guess tomorrow when the climbing season starts again, is using stable isotope analyses. Are adventitious roots actually uptaking mat water, so using a deuterium labeled water, watering the epiphyte mats and then looking to see that signature in the Sitka spruce leaves. So that should be pretty interesting. And then there's also the thought that maybe it's not adventitious root uptake of mat water, but it could be, not mutually exclusive here, it could be direct foliar absorption of fog and dew water through the stomatal pores. So using, again, stable isotope analyses, I plan to try to get to the bottom of that this winter. And then also kind of future directions here, take a peek at the fern physiology, so the vascular epiphytes. Here's the licorice fern growing on a branch. And there's a thought that maybe if the epiphyte mat was starting to dry out, there could be a new spin on sort of hydraulic lift. If you think about the host tree is wicking up groundwater high up into the canopy of the forest or the crown of the tree, and if the epiphyte mats start to dry out, it's possible that down a water potential gradient, water would leave the host plant through those adventitious roots and hydrate the epiphyte mat. So if I try to use stable isotopes, I could maybe see if in fact the ferns are getting any access to groundwater via their host tree, which would be pretty awesome. It would really redefine kind of epiphyte ecology, in the sense that epiphytes have typically been kind of classified as a commensal relationship where the epiphytes are gaining habitat expansion from their host, and the host is neither being kind of harmed or benefited by the epiphyte. But here we can see that maybe it's a lot more mutualistic exchange than previously given credit. Let's see, what else? Oh yeah, and then this winter I'll also be doing a thorough inventory of all of the epiphyte diversity on my Sitka spruce study trees. So all of the different macrolichens, microlichens, liverworts, and ferns. Should be pretty fun. With that, I would like to thank my collaborators, Nick Kerhoulas and Steve Sillett, who I've worked really closely with on this research. Also just HSU for having the infrastructure to do this research, and then Save the Redwoods League funded this research. And I think with that, I have some time for questions, maybe one or two. Thank you for your time and attention. Yeah.
Do you think you might see a seasonal variation of the curve that represents what you think is absorption because of the changing water in winter versus summer?
Yeah, yes. So I'm going to redo all of what I did last winter during the wet season, like next week, starting. So to do it in the dry season and see if there's a difference, because I think there probably will be a difference between the wet season and the dry season in terms of, in the dry season right now, those epiphyte mats are probably really dried out, and so that I'm expecting to find, but we'll see, and I'm not sure what I'll find, but that the water potential values in the host tree will be much more stressed, because I don't think that there's as much water available in those epiphyte mats, if indeed that's what they're uptaking. Does that make sense? Yeah.
Right, and you're looking at the tallest trees. Have you found any structural characteristics for when epiphytes start occurring for the adventitious roots, as far as height or size of plant?
That's a great question. It's kind of on my to-do list to try to do some sort of a map of kind of across the range of Sitka Spruce or Redwood about where the adventitious roots start, because it's a bit cryptic going around and finding them, because you have to disturb the epiphyte mats to kind of dig around and see if there is in fact, if there are in fact adventitious roots there. So as of yet, I haven't gotten a really good handle. Typically, the adventitious roots are only going to be on the really moist, like epiphyte loaded branches. I don't tend to see the adventitious roots on branches that are kind of naked, that haven't had like younger branches higher on the tree, like at the very treetop, that don't really have quite as many epiphytes there. They usually don't have adventitious roots, which makes me think maybe that on those branches that lack adventitious roots, that there could be some sort of direct foliar absorption of water. There's been a lot of research out of the Todd Dawson lab and stuff that's looked at different species capacities for foliar absorption of water, but Sitka spruce has not been investigated in that kind of crew of species that have been looked at. So this winter I'd like to, again, using stable isotope analyses, try to figure out Sitka spruce's capacity for foliar uptake. And the epiphytes don't get on the branches till the branches are pretty broad, kind of horizontally, like a little shelf sort of for things to get established on. If I do have extra time, I had extra slides to show you. I thought, so just to kind of give you some idea, this is from a colleague of mine, Alana Chin. She's been looking at the anatomy of the Sitka spruce to kind of go with my physiology investigation. And so what she's finding, I guess I should preface this with one thing that we think might be limiting photosynthesis. So in that Redwood picture that showed photosynthesis, photosynthetic capacity decreasing with height, some of my research showed that that's largely due to a denser mesophyll of the leaf. So all the cells in there, because they can't maintain turgor that well under hydraulic stress, they're kind of all jam-packed into the mesophyll, and that makes it really hard for CO2 to diffuse from the atmosphere to the chloroplasts and the sites of photosynthesis. And so what's really kind of crazy about these Sitka spruce leaves is that they have these sort of like channels or pathways. The mesophyll is organized into these kind of plates. And so I'm thinking that possibly this is meaning that you can have a denser leaf in the Sitka spruce a little bit, because there is some morphology gradient, but yet CO2 is still able, once it gets into the leaf, to kind of zip along through these open pathways. And maybe that's what's making it so that photosynthesis isn't quite as reduced in these higher density leaves. So that's one thing to show. So that internal, whoops, what did I do? That internal CO2 conductance is GI, and you can see it kind of decreases in the Sitka spruce, and that photosynthetic capacity there on the vertical axis is very tightly correlated to internal CO2 conductance. So the more easily CO2 can move through the leaf, the better photosynthetic capacity the leaf has. Probably enough time. I think I've satiated your interest in leaf anatomy and physiology. Okay.
One more question, or are we done?
Yeah.
Boy, that's a very similar leaf to the national park. It's very tall. So there don't be any.
Well, the two species are definitely closely related, you know, in the same family, and there is in redwood, clearly in coast redwood, due to that huge morphology gradient, there is a lot of plasticity in the redwood genotype for variation in leaf morphology. There's in the giant sequoia, there's not there's still a morphology gradient, but nothing like the champion leaf morphology gradient that's seen in coast redwood. That's kind of like takes the cake for the biggest change in morphology within a crown. Yeah, I think that there's two things that largely contribute to that. There's a hydraulic component, definitely, and then there's the difference in light environments. And it was kind of duked out for a while, you know, which was the leading mechanism causing this gradient in morphology. Some of my master's work used treetops of redwood at different heights, so they were all really high light environments, and there was just a difference in height, and you still saw that gradient in morphology, lending credit to, yes, there's a large hydraulic component.
Thank you.
Okay.